Can you see the screen?
Yes,
Yes.
we can.
Okay. First, I think that we'll get over with the book to be read in the November for December. Rama has gone the Russian focus and he gave us 3 options. And The Gentleman in Moscow by Amor Tolls is by far got a 60% votes, and then that's the book we are going to read. And we already had Canvassing by Trivi and others. That is a good book. So I think we'll enjoy that.
Yeah, we should have expected this book to be chosen, given that Rama was a gentleman in Moscow some years ago. So,
I don't know about the gentleman part, but I agree that the rest of it was right.
What we have not seen, we don't know.
Exactly. What happens in Vegas?
Rama, where's the stash of gold doubloons?
okay.
It's being used for the Ukraine invasion.
Okay.
Can you hear?
Oh, okay.
Yeah,
Yes.
we can hear. Yes.
Contains your genetic code, DNA, a molecule that makes up all your chromosomes. Double helix structure, a 2-stranded spiral. Set of instructions for your cells so that they know what to do, like send their signals and make new bonds. Making proteins from acid to amino. Complete set of genetic instructions is a genome. Nucleotides like genes and half the things that they're composed of phosphate, nitrogen base, and deoxyribose. Each nucleotide base is made up from these four, containing guanine, thymine, cytosine, and that's all. A always pair with D, and that's the base pair. C always pair with G, and that's the way they go. Deoxyribonucleic DNA is deoxyribonucleic acid, yeah. Deoxyribonucleic DNA is deoxyribonucleic acid. These complementary bases, each on opposite strands. You can remember the pairs, but the mnemonic GCAT, the control complex process to make a protein. From a gene, it goes transcription and translation to interception. The journey transcription takes information, transfers to mRNA messenger, moves the nucleus and onto the next stage. This is called translation. Happens in the cytoplasm. Ribosomes read the code on tRNA, makes proteins 1 amino at a time. Deoxyribonucleic DNA is deoxyribonucleic acid, yeah. Deoxyribonucleic DNA is deoxyribonucleic acid, yeah.
Very appropriate.
Wow.
Thank
Super.
you. Wow.
Very nice.
Okay. We have a special guest speaker today, and you have all seen Smita's very impressive CV. But just very briefly, to capture the route she took to working with CRISPR-Cas9 technology at CRISPR Therapeutics, and more recently at Takeda as lead of their liver rare disease team. She received her bachelor's in chemical engineering with minor in biomedical engineering, followed that with a master's at Johns Hopkins in chemical and biochemical engineering, and finally her PhD in chemical biological engineering and postdoctoral work in surgical oncology at Massachusetts General Hospital, Harvard Medical School. As if these exhausting and impressive academic credentials and work experience were not enough, Smita is a classical violin player in the Carnatic music tradition. We are really fortunate to have Smita, who is, as all of you would be glad to know, our own Ravi Shankar's daughter-in-law, and joining all of us at this book club session on A Crack in Creation. She is very gracious in agreeing to play the violin for us to set the mood before talking with all of us. Please join me in welcoming her to our book club today.
Certainly, certainly.
Thank you for having me. I'll just start with a short kriti. We just finished Navaratri, so this is the kriti Hiranmayim, a masterpiece of Muthuswami Dikshitar in the ragam Lalita.
Thank you very much. That was beautiful.
Excellent.
Beautiful.
Very nice.
Excellent.
Very
Really,
nice.
really. Super.
Thank you. Thank you.
Move to my table.
Mhm.
So, should I just start?
You can share, Smita.
Okay, yeah, one second. Can everyone see my screen?
Yes.
Okay.
Yeah.
So, I put together very basic slides, and before I start, I apologize in advance. I've been sick, so this is not my normal voice. So I'm gonna go through some of the just background to gene editing, in particular with a focus on CRISPR-Cas9 systems. And then I want to delve a little bit into the socioeconomic and ethical questions that arise when we think about CRISPR and what gene editing could mean in the grand scheme of humanity. So I titled this slide, I titled this presentation as Science Fiction Coming to Life because, you know, if we think back to Steven Spielberg's Jurassic Park, right, this is what he was saying, being able to engineer DNA so that we could recreate things that we no longer have. And so this is a real life example of something like that. And so for me as a scientist, this is extremely exciting, especially being someone who gets to work on these types of things. So before I start, I have disclaimers. I am not a molecular biologist. I am a liver biologist by training. My PhD was on metabolic liver disorders and the microbiome. My postdoc was on primary carcinomas in the microbiome. And then I somehow landed at CRISPR Therapeutics, which was, if you don't know, there were 2 individuals responsible for 3 if you count, the Broad, but Jennifer Doudna and Emmanuelle Charpentier were the 2 women who won the Nobel Prize in 2020 for the discovery of, they won in chemistry for the discovery of Cas9. And so Emmanuelle Charpentier is one of the founders of CRISPR Therapeutics. So that's where I worked. So, but again, I'm not a molecular biologist. I have not worked on the molecular evolution of these proteins. A lot of my work was translational. So the molecular biologists on the team would generate the, test drug products. And my job was to translate what it would look like in a model, in a disease model or in a non-human primate, and how that would translate to the clinic. The second piece is that, as I mentioned, I started under sort of the Emmanuelle Charpentier umbrella. So I have a very different perspective on the contributions of the 2 women. And of course, like equally fantastic, very talented, brilliant scientists. But so that is always that piece to keep in mind, that there is a little bit of like the politics, so to speak, between the 2 of them. And then the third disclaimer is that I haven't read the book. I told Ramarao uncle and Milind uncle that I would try to read the book, but I haven't had a chance to read the book. So I'm talking from a completely outside perspective. I'm not going to touch upon the book at all because I have no right to do so. I haven't read it. So yeah, so these are my 3 disclaimers. My 4th disclaimer being it's past 11 p.m. at night and 8 o'clock is my bedtime. So if I say anything wrong, please correct me.
Don't worry, you won't say anything wrong.
So I love this diagram because I think it does a really great job of explaining on a very basic level how CRISPR works. So CRISPR stands for clustered regularly interspaced short palindromic repeats. It doesn't matter what CRISPR stands for because to most of us don't even remember what it stands for. And most of us don't even understand where these words came from. So don't worry about what CRISPR stands for. What is really, really important here are 2 components. The first one being the Cas9. So this is the component that Jennifer Doudna and Emmanuelle Charpentier won the Nobel Prize for the discovery of Cas9. And Cas9 is ubiquitously found in many, many, many bacterial species. And the reason for this is that it is actually a defense mechanism. It allows them to cleave foreign DNA from a plasmid or a virus or a bacteriophage. And so in that sense, it actually is meant to destroy DNA. And so when we understood how bacteria can cut DNA, we harnessed that protein. We isolated that protein. And the first one that was isolated was from Pyrogenes, which is the typical canonical Cas9. And so that Cas9 protein complexes with something called a guide RNA. So the guide RNA is what tells the Cas9 protein where to cut. And so immediately adjacent to that guide RNA, it's a 20 base pair sequence for the canonical Cas9. Adjacent to that, what we call the spacer, is something called a PAM. And so that PAM is actually the sort of the key, the target ID. It's the way that the Cas9 and the guide RNA know that to bind there. So without that PAM recognition, the Cas9 cannot bind. And so that PAM changes depending on what type of Cas you're using. So for a canonical Cas9, the PAM is NGG. So you guys heard the wonderful song in the beginning where you learned the 4 bases. If you didn't already know, the 4 nucleic acid bases that are found in deoxyribonucleic acid. So we have guanine, cytosine, adenine, and thymine, and these are complementary bases. So guanine can only pair with cytosine, and adenine can only pair with thymine. And so the NGG PAM recognition site means that you can have any one of the 4 base pairs followed by 2 guanines. So that is the PAM that is recognized by Cas9. And so that tells, between that PAM and the guide RNA, that tells the Cas9 where to cut. And where it cuts is 3 base pairs upstream of that PAM. So for example, if you have a 20 base pair guide RNA and 3 base pairs of the canonical NGG PAM, the guide RNA would cut 17 base pairs into the guide RNA because it cuts 3 base pairs away from the PAM. So I hope you guys followed that, but it doesn't matter if you didn't. And so then once the Cas9 cuts, it causes a double-stranded break. And please remember the double-stranded break because I'm going to talk about the evolution of CRISPR-Cas9 systems on the next slide. And this distinction between something called a nick and a double-stranded break is very, very important in this evolutionary discussion. And what happens after that double-stranded break? So 3 things can happen. You can insert a base, you can delete bases, or you can do nothing. So these are the 3 options. And typically, various cell types have various repair mechanisms. So Cas9 relies on the repair machinery of the cells to be able to repair the pieces of DNA that are now fragmented. And that repair machinery is not perfect. And the repair machinery, depending on where the guide RNA has broken the DNA, has different levels of imperfection. So for example, one site may never cause any sort of insertions or deletions. It may repair perfectly every time. One guide RNA may cause 1 base pair deletions consistently, meaning 90+% of the cuts cause a 1 base pair deletion. Another one may cause 90% plus cuts to have a 1 base pair insertion. So these are entirely dependent on the cut site of the guide RNA and the repair machinery of that cell type. So even if you can find the same locus across different cell types rely on different types of machinery depending on how they divide, because it is the dividing cells that have a certain type of machinery. And for example, hepatocytes, they don't divide. They're non-dividing matured terminal cells. And so they have a very different repair mechanism. So I won't go into the repair mechanisms because that is a whole different discussion, but that's just really, really important to consider in terms of the double-stranded break. So this is the schematic of how CRISPR-Cas9 works. And so before I go on, does anyone have any questions on this? Did it not make any sense at all?
Oh,
Okay.
you are very clear.
No, it was very, very clear.
Very clear.
Thank you. Okay. So 90% of the time when I explain science to people, I say the science isn't actually relevant to having the ethical and socioeconomic debate. But in this particular case, with regards to gene editing, the science is very, very important. And you will understand later once we get into that part of my talk. So now we're into, the next generation iPhones, right? So we have CRISPR 1.0, which is our double-stranded break, which I just explained. It's the standard canonical Cas9, which uses a single guide RNA to cut and cause a double-stranded break in the DNA. The CRISPR 2.0 is something called a base editor. And so this came out of David Liu's lab at the Broad Institute. And so what they just said was, okay, the double-stranded breaks are causing a lot of problems. And this is something the FDA really doesn't like. They don't like the fact that nucleases cause double-stranded breaks. So what they did was they engineered something called the Cas9 nickase. And what the nickase does is it will only nick 1 strand. And this is why the double-stranded break was very important to remember. So this version of CRISPR can only nick a single strand of the double helix. And so how it works is, if you look here, the guide RNA binds and causes a nick. And then the deaminase comes in and it can convert either an A to a G or a C to a T. Okay? So it can only do those 2 conversions. It cannot do anything else. And so this is a really nice tool to have in your wheelhouse for specific diseases that have, so for example, in alpha-1 antitrypsin deficiency, there is a single mutation that causes, it's called the PiZZ mutation, where 90+% of patients have the PiZZ mutation, and it actually is an A to G, it's a G to A transversion. So this is a beautiful system that doesn't cause a double-stranded break, which means you can't have any insertions or deletions, and it will change only that 1 base pair. And so this was a great finding. This was something that was absolutely magnificent, and Beam Therapeutics came out of this single technology. And so that this was a really important sort of hallmark, like sort of time stamp in the CRISPR-Cas9 community, because it really taught us the importance of differentiating between a double-stranded break and a nickase. And then what happened was David Liu went back and said, okay, we did the base editor. Can we do something that can do anything? So the base editor can only do a C to T or an A to G transversion. Can we design something that can do anything but using a single nick? So what they did was they took the nickase, again, the engineered nickase, and they fused it to a reverse transcriptase. And again, going back to that song that we just all heard, reverse transcription is when you take DNA or RNA and you reverse transcribe it back into DNA. So now you provide something called a prime editor guide RNA, which we call a PEG. And so the PEG does two things now. The PEG tells the Cas nickase where to nick, and it also provides a reverse transcriptase template. And that RTT can now introduce any change you want into the DNA. By reverse transcribing from that template that you provide, you can now insert or delete or make any changes within that sequence that you want. And then when the second strand is synthesized, it is synthesized based on the first strand that you have now edited. So there's a lot of engineering that goes on in the base editor. It was the prime editor, which I'm not going to talk about. There's ABE8. I think they're on version 8 or 9 right now. And then the prime editor, we're on PE3 or something like that. So these proteins are constantly going through the molecular evolution to make them better, to reduce the off targets, to increase the efficiency, changes in delivery, looking at AAV versus non-viral delivery systems. So a lot of those things are happening, on the peripheral, in the periphery of the grand scheme of this. So I really like to, tell people about these different systems, because this changes the things that we can do. It expands the things that we can do. And so from an ethical perspective, yes, you can cure more diseases, but if you don't draw that line, what are the other things that you can do? And that can cause harm. So that's just something to keep in mind with regards to these engineered editors. In terms of, where we are in terms of what we have shown proof of concepts for with regards to gene editing. And so far, we have shown proof of concept for anything under the sun. We have shown that you can use a standard CRISPR-Cas9 system to splice out multiple repeat expansion disorders. So for example, Huntington's, ALS, Parkinson's, these are all caused by something called repeat expansions, for Friedreich's ataxia. And so with 2 Cas9 systems and 2 guide RNAs, now you can cut out those repeat expansions, and you have a way to tackle repeat expansion disorders, which we never had before. We've been able to show that we can actually make our CD34 or hematopoietic stem cells churn out fetal hemoglobin. So patients who have beta thalassemia or sickle cell anemia, they now can have fetal hemoglobin so that their blood cells are functioning properly, and they are able to not sickle, which is where the term comes from. We've used it in the context of cancer. So we've engineered T cells to be able to better fight off solid tumors, lymphomas, anything stemming from, in terms of melanomas, primary hepatocellular carcinomas. We have shown that we can engineer these cells to be better at attacking cancer without a chemotherapy. And then we've also shown it in the context of metabolic liver disorders. We've been able to engineer things and directly inject into the eye to deal with cataracts and retinal degradation disorders and cystic fibrosis. So the proof of concepts are wide reaching. It's quite impressive since since we discovered the Cas9, how much proof of concept we have generated. And we've also put a lot of things in the clinic. So Intellia has shown that they can knock down TTR for patients who have cardiovascular diseases due to the excess cholesterol. So that was the first proof of concept that we could edit in a patient. We could directly inject CRISPR-Cas9 with a guide RNA and knock down a gene. We've shown that we can actually synthesize, we can engineer hepatocytes to synthesize factor VIII, synthesize and secrete factor VIII. So that's been amazing to see. Again, we've shown this in the context of beta thalassemia and sickle cell, which was actually a very nice story that I like to share with people. The first patient dosed with a CRISPR-Cas9 system, ex vivo edited cells put back into a patient for beta thalassemia, the patient was dosed on my first day at CRISPR. So my first day at CRISPR, they handed out AirPods to everybody. And so that was my first day in industry. I was like, hey, this is great. It's way better than academia. I made the right decision. So it's absolutely phenomenal how much we've been able to do with CRISPR-Cas9 systems. And I'm happy to talk more about this if anyone's interested in terms of the applications and the proof of concepts that have been generated. But we also have a lot of challenges. So, for example, we have a lot of issues with delivery. Delivery is the most difficult thing, not just in CRISPR-Cas9 systems, but in monoclonal antibodies, in vaccines, in small molecules, any type of therapy, delivery. We always have this saying in any company that you need to solve delivery, And the reason is, and as a translational scientist, this is really important to me because when somebody tells me that something works in cells, I don't believe it. I don't believe that something's going to work until I see it happen in a non-human primate. And those translational studies are all based on our understanding of being able to deliver everything. Deliver to the target tissue, deliver with the right amount of efficacy, not have something lingering around, especially something like Cas9. We don't want it lingering around for years and years because who knows where it's going to cut after that, right? So there's a lot of safety and actual efficacy questions that we need to be able to address in terms of the delivery perspective. The other big one is off target. So now you have a guide RNA, and I didn't really talk too much about this, but the guide RNA is chosen based on its homology to a single site in the human genome. But what is really, really, really interesting about Cas9, and I talked about this PAM, right? The cut site is three base pairs from the PAM. The further the mismatch is from that cut site, from the PAM, the more likely a guide RNA can cut at an alternative site. So this is something that's really, really important. So the off-target, our understanding of how much off-target editing we have, that's really, really a challenge, trying to get guide RNAs to only cut at the locus of interest and not anywhere else. So these are all things that are also part of the engineering that we do for the Cas. For example, we developed something called a high-5 Cas9, which is called a high-fidelity Cas9, which is much more dependent on that homology being perfect. And if the homology is less than perfect, it is less likely to cut at an off-target site. So these are also growing concerns, especially with regards to the FDA and with everyone's IND applications and filings and everything that's going on in terms of where the field is moving, right? So these are not questions that the FDA has ever had to answer. So these are evolving discussions, and we're learning more and more about how to understand off targets and how to predict them and how to better mitigate them. And then the autoimmune response to Cas9 protein, right? So we're putting something foreign in the body. You're going to have an immune response. What does that immune response look like? And what happens if you have an AAV delivered Cas9 that sticks around for a long time? Are you now going to have an immune response to that Cas9? So these are really, really, really important. And then of course, gene polymorphisms that can cause cancer, right? You're editing the genome. We don't know what the long-term implications of that are, right? And so these are all really, really important challenges that we always have to keep in mind. And these are, again, evolving discussions that we are having in the gene editing space about how to tackle them, how do we mitigate them, how do we predict for them. This is where computational tools are becoming more and more important. And how do we regulate them, right? So those regulatory questions are still in discussion and in play. And Rama Ralanka will have a lot more information from the clinical perspective on this, I believe. So I think he would be a good person to ask about that stuff. And then, of course, the CRISPR-Cas9 ethical issues. So before I go into the ethical discussion, I do want to touch upon these two things. So we're talking here about, somatic cells versus germline cells. And this is where the ethical questions arise, right? So somatic cells are cells that are already terminally differentiated. So your blood cells, your liver cells, your pancreatic cells, your epithelial cells, your intestinal cells. So all of those cells are somatic cells. They cannot pass any sort of edits or into offspring, right? The only cells that can do that are sperm and egg cells. And so now the question becomes, if we can target sperm and egg cells and we can target germline cells, what does that mean from the perspective of autosomal recessive and autosomal dominant disorders? But what does that mean in terms of where we draw the line between something that is actually an issue versus something that is a desired trait? And so this is the part where I want to sort of engage everyone to think about all of the things that I've talked about in terms of what we could do with a CRISPR-Cas9 system. So really imagine what we could do. We could engineer food, right? Food shortage is such a big thing. And if we could engineer plants so that they grow faster, so that they're hardier under drought conditions, for example, they don't need as much water, they don't need as much sunlight, they don't need the high temperatures, they do well in the cold, we can generate more crop in the cold. How amazing would that be? And same thing, we're able to generate animal models now. CRISPR is used across the board to generate animal disease models so that we understand human disease a lot better. And we're able to generate humanized disease models, which is absolutely a phenomenal concept, right? We can put in a human transgene containing a mutation that a person would carry into a mouse and look at the phenotype. And while it's not perfect, there's so many learnings that come out of that. We're able to do engineering of drug delivery modalities, right? So if we can do a nucleic acid drug delivery system, you can engineer it however you want because you have the CRISPR-Cas9 to do it. And then obviously gene surgery. And that is coming from the perspective of being able to engineer stem cell derived organs. So if you could generate hepatocytes and be able to do an orthotopic liver transplant instead of needing a whole liver from a healthy donor or partial liver from a healthy donor. So we no longer have to do partial hepatectomy from a live donor. We no longer have to worry about HLA matching because you're taking the patient's cells and you're putting the patient's cells back in. And so these are the opportunities are absolutely endless. And I didn't mention fuel, right? We could, if you're using bio-based biofuels, you can engineer them because again, all living organisms have DNA that can be edited. But then think about what we shouldn't do, right? So this is what I was saying when we're talking about the germline editing, right? What is actually something that a child shouldn't have to live with versus something that we don't want our children to live with, right? That's a very difficult line to draw. And something like, some people could argue that like low levels of autism is actually not a bad thing. Autistic children often have very, very unique traits. They have very unique capabilities. They often bring something so different to the table in terms of discussions. But is that something that we should be correcting in children? Same thing, there are many, many disorders that, dwarfism, is that something that's actually a problem? These people live pretty normal lives, but do we want a child to struggle with dwarfism? Is it a struggle? So these are really, really important ethical questions that we absolutely need to be thinking about. And they're really hard questions. I don't think there is a right or wrong answer. I think it's, as a society, what do we think is good, okay to have? What do we think is actually bad to have? Is the line drawn at, oh, if someone's life expectancy is a little bit shorter, that's where we draw the line. Or if someone's going to live a more difficult life, if they struggle to achieve the same things as other people, is that where we draw the line? And so as a society, this is a very, very important question. And it's an important question for the FDA, to be honest, because at some point, companies will start sort of venturing into those areas without a doubt, because, scientists are curious. Scientists want to solve everything. And I think that this line needs to be drawn sooner rather than later, before those, sort of riots crop up. But just something, to leave you all with in terms of what we should be thinking about. And then on top of that, I just wanted to touch upon some other considerations that we should again be thinking about. So the cost of some of these CRISPR ex vivo therapies, these CAR Ts, the CD34 engineered cells that have been put into the beta thal and sickle cell patients, they're extremely expensive. These are on the millions and millions. They're on the order of millions of dollars. Most people cannot afford that. Insurance companies, we had a whole department at CRISPR that was dedicated to spending their entire day talking to insurance companies, trying to get them to somewhat subsidize some of this, but they won't because it's too expensive. And then along with that, accessibility, not just in terms of cost, but there are people who don't even have access to a normal clinic. They're being treated in a tent somewhere by a doctor or surgeon who actually doesn't even have sterile tools. Accessibility physically is also very, very difficult because, again, with regards to cost, it is very expensive to generate these medicines. Most people can't afford them. What are people going to do, in third world countries where maybe some of these diseases, for example, Wilson's disease, there is a mutation, there's the R778L mutation, which is actually very, very prevalent in Asian countries, many of which have poor populations who cannot afford a gene editing therapy for Wilson's disease. So again, accessibility is something really, really important. So long-term impact. What is the long-term impact of gene editing? We honestly have no idea. We don't know who's going to develop what cancer or what sort of genetic defect moving forward because we did something. Manufacturing is very difficult because scale up of something like a guide RNA is really, really difficult. mRNA, so our Pfizer vaccine and our Moderna vaccine, they were mRNA vaccines. And I had a colleague who was working as sort of a collaborator company on helping them do this scale up, and they struggled. The vaccine was actually ready way before we actually got it out, but manufacturing of RNA is so difficult. To get that purity, to be able to put it into a patient is extremely challenging. And then the two things I want to touch upon at the end, which some people don't think is a big deal, but I personally do. It's the loss of the cheaper, not so cool medicines and the loss of traction on other technologies. So there's this CRISPR craze going on. And so a lot of these companies that were working on small molecules, they struggle. They're kind of still struggling because they're not doing, they're not dealing with the new kid on the block. They're kind of dealing with a little bit older technologies. But small molecules are cheap. Small molecules have done so much. We have so many amazing small molecule inhibitors. We have a small molecule aldose reductase inhibitor that works beautifully for galactosemia in preventing many of the severe complications from some patients that have clinical variant and classical galactosemia. And small molecules have been used in the brain. They have some small molecules have great BBB penetrance. And so these are things that we always need to be keeping at the back of our mind. And so that's where I wanted to just quickly touch upon. Let me know if I'm running over. I can stop talking at any point. But in my job, actually, the nice thing about my current position is that I am completely modality agnostic. My job is to look at a disease, look at an indication and say, this is the best technology for it. This technology will not work. This is not the right technology for this particular disease. And that is a really important sort of decision-making process that many of these smaller biotechs that have a single technology have lost, because that is their bread and butter. For example, when I was at Prime Medicine, Prime Editing was the bread and butter. When I was at CRISPR was the bread and butter. You couldn't do anything else. But at Takeda, I've been able to work on non-viral DNA technologies. I've been working on small molecules. I've been working on standard AAV-based gene therapies. I've also been able to work on gene editing. And so thinking about a disease and understanding the molecular mechanisms of that disease, and then making a very data-driven decision about what technology is best for that disease and that for that patient population is key also. Who is the patient population? So, right, we would never, gene editing in children it's a hard sell. It's a really hard sell in children, but a small molecule is not, right? And so we always need to be cognizant of the patient population as well when making these decisions. So with that, I will leave you guys with a few additional of books that some of which I have read, some of which I've skimmed, but I highly recommend The Double Helix. If you have not read The Double Helix, for whatever James Watson was as a human being, the book is worth a read, because the double helix understanding of DNA structure is what allowed all of this other stuff to happen. And so that's a really important basic foundational book that I think is absolutely worth reading. And then The Gene, again, Siddhartha Mukherjee, fantastic, actually book recommended to me by my father-in-law, Mr. Ravi Shankar. So he was the one that told me to read this book. So I credit him for that. And then the last one, Being Mortal, again, recommended by my father-in-law. So just understanding where we are ethically in terms of gene editing, what we should be doing. And also the nice thing about Being Mortal is that it really talks about medicine from a holistic perspective in terms of at what point do we just say no more, right? And we tend to forget that quality of life is so important, and that quality over quantity in all aspects of life is what we should be aiming for. And so I really like that book as well. So that's all I have. I'm happy to take any questions that anyone might have.
Thank you so much, Smita.
Yeah,
I think you've been an excellent teacher. And I say that because making such complex materials simple is not an easy task. And with your clarity and with your detailed, beautifully done slides, you accomplished that. So thank you so much for taking the time. I'm going to just have a couple of questions for you from my friends, and then we'll let you go because it's getting late in the hour for you. Okay?
Yeah.
Okay. Yeah,
I do.
Go ahead, Raja.
Yeah, I do have one question. I mean, that is, first of all, that is a brilliant articulation and explanation that is provided by you. I mean, it's just amazing. So thank you for that. The question that I have is, why is the subsequent work on this still at the proof of concept stage? One would think with the immense potential that this offers, that there would be a massive amount of investment and activity and progress, therefore, to bring it to a point where it's actually being applied in at least one of those disease areas to a much greater extent than just remaining at the proof of concept level. So what's hindering that progress?
So there's a few things. The first one being the two components that I mentioned, delivery and the off targets. So off targets is a really big issue. So what happens, there's an irony, there's a little bit of a catch-22 when it comes to guide RNA. So I mentioned to you that different guide RNAs have different, they cut at different sites in the genome. And the DNA is protected by something called chromatin. And so local chromatin structure at different points in the genome can cause different accessibility issues. Physically, like sometimes you cannot access the genome. And so that's why we can have 10 different guide RNAs at the same gene that have 10 different cutting efficiencies. And so what we will find more often than not is our lead guide RNA that cuts really well at our locus of interest also has a really bad off target somewhere else where it cuts a lot as well. And so that is the biggest, it's a catch-22. So the guide RNAs, just because they cut well at the locus, it's going to mean that they're going to cut somewhere else as well. The second piece to that is the delivery. So with regards to, for example, anything that needs to cross the BBB, we do have AAVs that can cross the blood-brain barrier. We have things like transferrin receptor sort of signaling tags that we can put onto proteins to have them being taken up by certain cell types, for example, neurons. But crossing the BBB is still a big challenge. To deliver enough to actually make an impact is very, very difficult, because you also need to be very careful about the fact that when something goes into the brain, it's going to have a lot of trouble getting back out. So you cannot overload the brain, especially with AAV. We do not have non-viral technologies that can penetrate BBB right now, or at least have the penetrance that we need. And the third component to that is low-hanging fruit. So most companies started with the liver. So you take a Cas9 mRNA and you take a guide RNA, you throw it into a lipid nanoparticle. Where is everything going to go when you put it into a human? It's going to go straight to the liver. And so most of these companies started with either ex vivo editing. So CD34 cells are really easy to take them out of the patient, you engineer them and you put them back. So that was the first low-hanging fruit. And the second low-hanging fruit was non-viral delivery to the liver. And so if you'll notice, a lot of the proof of concept are blood-brain barrier issues and extrahepatic tissues that are really hard to deliver to. And then the guide RNA off targets.
Thank you. Is there one more question that someone would like to ask Smita? Okay. Thank you so much for your presentation.
I have a question, Milind. If we have a minute, can I have one question?
Sure.
Smita, you mentioned something very interesting towards the end, that quality and quantity of life are really a question. In being mortal, and you came out very strongly on the quality of life, which is something I completely agree and believe in. In Atul Gawande's book, In Being Mortal, he actually at the end says that his father always believed in quality of life, but at the fag end of his life, when...
Chacha, can you mute, please?
But at the fag end of his life, Smita, in the book, Atul mentions that his father was actually incapacitated, but didn't want to die and asked for his life to be prolonged. And he said, so even the things that we decide when we are healthy and the way we think we're going to feel, when it eventually comes to the crunch, even a father as committed and as clear and as energetic as his own father changed his mind. So I was just wondering if you have any comments, considering I completely side with you on quality of life. So any comments on it?
So I'm speaking with a lot fewer years on the earth than others. So that was just my take on the book, because in terms of the ending, yes, you're correct that he didn't. When it gets to that point, most people don't want to let go and they don't want to move on and pass on. But one thing that I always, so one thing my mom always used to tell me, that she's like, I always go to the gym and I always work out and I'm trying to eat healthy. And she's like, I don't do it so that I can live longer, right? I do it so that I never become a burden on my children. And so that's always something that stuck with me in terms of like living a really good life and living to the point where you and your family are happy. But beyond that point, when you get to the point of other people having to sacrifice their quality of life to take care of you, that's sort of that was sort of my sort of crux, that my issue with that piece of the book. But yeah, that's my 2 cents on it. Just I can only pass on the wisdom from my mom.
Thank you. That was that's very thoughtful. Thank you.
Yeah. Well, you've really done justice to both the questions, and there are lots of lessons to take on what you have presented to all of us. So thank you again.
Thank you for having me.
Have a good evening.
Yeah. All right. Bye.
Thank you.
Bye.
Thank you so much.
Thank you so much. Thank you. Bye-bye.
That was really impressive. Huh?
Yeah.
Anjali,
Excellent.
Ravi Shankar, take a bow. Yeah. That was very impressive. We're going to give you a lot of credit. I don't think I can persuade
Yes.
my daughter to do a session for me. Yeah? You persuade your daughter-in-law. Let's give Ravi a pretty round of applause. Yeah? I'm damn impressed. Very impressive. Sorry.
So,
Go ahead.
yeah,
Ravi
Anjali,
Shankar's got two very smart daughters-in-law. You've just seen one. The other one is also equally brilliant. That is
Great.
fabulous.
Okay. So let's move on to the main part of our program. Zareen, as our Reader of the Month, has worked really well with her team of reflectors, Raja, Vinita, and Nidhi. And for the cutting edge book that she has recommended to all of us, she will now offer an overview that sets the stage for all three reflections that follow. So please go ahead, Zareen.
Yeah, thank you, Milind and Eranki. In fact, after hearing Dr. Krishnan, I'm wondering whether an overview is really necessary, because she's done such an amazing job that even a layperson like me could understand the book better. So Eranki, I must say that compared to the video that you showed at the beginning, the book is very different. I found it to be an intense read, quite academic in terms of its writing. But let me very quickly highlight what excited me about the book, which is, as Dr. Krishnan also mentioned, what is the potential that this technology really offers? So if we see, as late as last year, in 2020, there were 241 million cases of malaria, which led to 500,000 deaths. This is just last year. Imagine a world where mosquitoes lose the ability to carry this disease. Imagine if there is no malaria carrying mosquito. Similarly, even today, 8% of the world's population suffers hunger and food insecurity. 8% of people in the world go to bed hungry and have food insecurity. Again, picture a world in which staple crops are resistant to droughts and diseases, and the pressures of climate change don't affect them, which means there is bumper crops for all of us to have. Food is never a shortage. Similarly, identifying, and Dr. Krishnan alluded to this, I think, in a talk, identifying and correcting just one gene out of 3 billion genes can cure sickle cell anemia, which is a disease which is still prevalent very largely in Africa. Now, these may look like fantasy fiction, but this is what's possible thanks to CRISPR-Cas9, which was discovered by Jennifer Doudna and her colleagues. Yeah, the next one, Eranki. Doudna writes in her opening introduction that we as humans have always been very interested to change the physical world. Industrialization was the first phase that we went to, but we now realize that it brought climate change and species extinction. Where are we now on? We are at the last frontier that we are currently working on is technology. Whether it's AI, space, or biotechnology, these are the areas which we are now trying to master. And with biotechnology, and this is really relevant, the controls of who we are, what we look like, how we behave is no longer left to evolution, but to a simple science of genetic scissoring. So there is so much of power behind this technology that it's sometimes really scary. The next one. I'll just quickly read through this because she explained the entire CRISPR-Cas process really well. In 2012, Jennifer Doudna and Emmanuel Charpentier came up with probably the most important biological breakthrough, which is a tool for gene editing. They discovered a bacterial immune system, which is now known as CRISPR-Cas9, a sequence which reads the same forwards and backwards. And what is CRISPR? It is a technology derived by studying how bacteria have been successfully inhibiting virus from invading it. It's basically a bacteria's own immune system. How does a bacteria prevent a virus from invading it? That is what was discovered to be the key behind CRISPR. And CRISPR can be reprogrammed to cut any DNA from any organism, sorry. More than the tool, again, like I said at the beginning, I found the applications really fascinating and troublesome, both. So already in science labs, and again, Dr. Krishnan's talk highlighted this, scientists are working on genetically enhanced version of the small beagle, dogs with supermuscular physiques, micro pigs. We are also working on crops which are more resistant. Next, please, Eranki. Plant genomes which are improving people's diets, disease-resistant rice varieties, tomatoes that ripen slowly, soybeans that are healthier with polyunsaturated fats. So even our entire food is getting reinvented. Scientists are using CRISPR to detect specific targets, DNA from cancer-causing virus. And most importantly, the impact of gene editing on the human species is the most exciting. CRISPR has been already used to humanize the pig DNA so that they can serve as organ donors for us in the future. Mosquito strains are being worked on so that mosquitoes lose their ability to carry disease and therefore mosquito illnesses can be eradicated. All this is actually happening in the labs as we speak. But there are some ethical issues, and those ethical issues really are questions which we all need to ask ourselves while science goes on this rampage of discoveries. And what are this? One is that DNA of future generations can be also altered. So imagine a world where parents can tailor-make their future offspring so as to ensure that the child has no congenital issue or faulty genetic lineage. Or imagine also a parent specifying the physical, mental, emotional characteristics of their child to be born. It's a little scary. With knowledge and power, people who have knowledge and power can dominate what it's called gene drives. And here Dubna herself says that she was repeatedly plagued by a dream which she used to get, where Adolf Hitler comes to her and asks her that I want to understand the uses and implications of this amazing technology you've developed. Imagine the ability to make mini Adolfs or mini Musks or mini Zs. It's possible with CRISPR, but is that what we want? Also, it's very important that mainstream media covers these scientific discoveries. Today, if you see, mainstream media focuses a lot on political and economic issues, whereas scientific discoveries are not mentioned. And this in turn makes common people, lay people like us, unaware, uneducated. And therefore, we are not able to debate or discuss the right and wrongs behind all of this. So that's another point. Yeah, last, please. For all the possibilities that CRISPR offers, there is an underlying dose of caution. And Jennifer Doudna herself says that it's not the technology which is good or bad. It's what's important is how it is used. So I think with this very quick overview, I'd like our three reflectors who've got so much of material to talk about to explain the book further. Thank you.
Thank you so much, Zareen. That was an overview, really well done. It shows us what are the challenges facing CRISPR-Cas9, as well as the challenges facing novice readers like me, who are not fully informed. Those challenges are apparent. I, of course, learned a lot from reading the book. Now, our reflectors have taken on the challenge far better than I would have. And in reading the book, you will see that they are coming up with some very useful and valuable reflections. So let's start with Raja, who will speak to the first half of the book that was called The Tool. Go ahead, Raja.
Thank you. Really hard acts to follow. Are you able to hear me?
Yes.
Okay.
Yeah.
So those are two hard acts to follow. So, I'm glad that Dr. Krishnan is not here to listen to my explanation of the tool, because I'm sure she would have had to bite her tongue. So let me start off with asking the question, where were you on Friday, June 8th, 2012? I distinctly remember where I was. I was discovering some new wine with my family at a winery that we were visiting somewhere in Georgia. So where were you? And this was the day that this fairly simple, innocuous looking sentence was kind of released into the media out there, into the scientific community, which essentially said we propose an alternative methodology based on RNA programmed Cas9 that could offer considerable potential for gene targeting and genome editing applications. And it was really, that's the day when the world was first informed of this powerful, mind-blowing new era of genetic command and control. So this book is the story of how it came about, of the power it unleashes, and the enormous responsibility that it now requires from all of us on how that power should be used or should not be used. I, for one, don't believe that a clear line can ever be drawn around those issues. So we'll have to see how it evolves. It's very rare indeed for someone in the deep scientific research community who was central to a discovery of this magnitude and enormous potential with, as the title itself says, the unthinkable power to control evolution, to write a first-person account of the journey, how it all came about, the aspiration for this holy grail, the seeming impossibility of being able to discover how it all works, the independent research efforts, the toil, the genius minds involved, the accelerated advancements that result from an entire global community building on top of each other's work, the fortunate strokes of serendipity that opened up new trails to pursue, the race, the thrill of knowing you're onto something big, the methods used, the focus of the small collaborating team, very small team, the final stage of the hunt, and the joy of discovery with not only acquiring a clear understanding of how it all works, but inventing how you can take command of that process to precisely control the outcomes. And finally, the feeling of excitement and euphoria in the moment when the author presses the send button on June 8, 2012 to inform the world about it. If we can go to the next slide. So the book is an amazing account that chronicles the entire path to final discovery and the connectors feeding into it from other amazing work done over many decades. It provides a glimpse into the people involved, their motivations and contributions, the milestones reached along the way, each of which was rewarding at the time it was achieved. Interestingly, it also points out how important commercial research can be in contributing to fundamental scientific research. So many of the crucial stepping stone discoveries that fueled the eventual outcome were the result of commercially driven research funded by the dairy industry, in particular a company called Danisco, whose investment interests in the production of yogurt through milk fermenting bacterium called Streptococcus thermophilus were under constant threat of phage infection, which caused enormous production bases, losses and incomplete fermentation. What is astounding to me is how quickly it all came together from a meeting where the author, Jennifer Doudna, and by the way, did you see how, she had to be one of the chosen ones to find this? Her name ends in DNA. Where the author heard about CRISPR for the very first time, she didn't even know what it stood for. And that was in 2006, when she and her collaborators, until when she and her collaborators announced their discovery to the whole world just 6 years later. Sounds impossible, but it happened. And it will surely serve as an inspiration for all budding, aspiring scientists. So that's good from that point of view to inspire other scientists out there. So what exactly is it that was discovered and how did it come about? What is the tool? How does it work? What is its potential for enormous impact? And why is it so amazing and so powerful? Let's take a quick, a peek at the story and the outcome. So what I've done here is to try to, capture some essential milestones that came about. For one thing, in nature itself, over 100,000 years of Homo sapiens existence, genome has been shaped by the twin forces of random mutation and natural selection over this time frame. And during this time, there were certain, instances of gene editing that happened, not by design, but through chance occurrence. And that is equivalent of winning the genetic lottery. But, some of those instances really sparked the interest and the curiosity of scientists who wanted to try to understand better what was going on there. And in the 1960s onwards, you had, the concept of gene therapy, which is using viruses, essentially viral vectors, to deliver, and in this case, to insert therapeutic genes into a cell's genome, including in humans. And viruses have the uncanny ability to splice new genetic information into the DNA of bacterial cells. And then you had the concept of homologous recombination that led to gene targeting, essentially, gene editing. And some of that work ended up winning the 2007 Nobel Prize. And this essentially is based on what happens in nature in procreation with exchange of similar DNA sequences between one chromosome to another. And this is an important source, that process is an important source of why offspring don't always have 100% of the exact characteristics of its parents. And there's some genomic variation or genetic diversity that is introduced as a result. And this method was, in fact, it discovered it could be used in order to effect gene editing. But the success rate of this was just 1%, which meant that, the illegal recombination that is happening was about 99% of the time in a random kind of fashion, and only 1% that is targeted was successful. And clearly that is not something that's acceptable. More precise repair was introduced in the 1990s with the ISCEI endonuclease. Nucleus is what allows you to actually do the cutting of these genes, of the sequences. And the precise repair was possible of a mutated gene, essentially homing endonuclease. But this, again, had only a 10% success rate, still not acceptable, but exciting, though, in terms of development and progress. Then you had in the 2000s, the ZFNs, or the zinc finger nucleases, gene editing with custom-built ZFNs. Each finger recognizes a 3-letter sequence of DNA, but this, as well as the next stage of evolution of this discovery with tails, so the transcriptor activated like effectors, and the nucleases in combination with that called talons, which would recognize a more precise single given letter in the DNA. These are very expensive to create those custom ZFNs, or the talons. And so it is no wonder that when it came to this particular discovery in June 2012, the ability to edit not only the DNA of every living human, but also DNA of future generations, to direct the evolution of our own species, and do so at a much, much cheaper cost and in a much simpler manner, was something that is a mind-boggling discovery. And there were 3 critical requirements that had to be satisfied during the course of this that really didn't, all 3 didn't get satisfied, especially the third one, until this particular discovery came about, because it required the ability to recognize a specific desired DNA sequence, the ability to cut that DNA sequence, but more importantly, the ability to be easily reprogrammable to target and cut different DNA sequences. Some of the methods and mechanisms that were discovered earlier had to be done in such a custom way that it could only, at the end of that process, could only be used for a specific scenario. And if you had to do it for another scenario, you had to start from scratch. But this ability to be easily reprogrammable was essentially one of the most important aspects of this discovery. So if we can go to the next slide, and the last one. So, we all know what CRISPR is, and, I don't need to go into the details, and since, Smita explained this, so well. But essentially in nature, bacteria had found a way to program a warrior protein to seek and destroy viral DNA. And armed with a cache of RNA molecules derived from the CRISPR array, which essentially is the beauty of this, is that the CRISPR array, by definition, those palindromic repeats that you have, which are really the diamond-shaped symbols, the darker ones that you see on the top left-hand diagram there. And in between are the spacers, which really is the memory of previous infections that the bacteria had that it saved a copy of. And so the Cas9 was able to combine and create these the structures that could then be deployed to search for and find equivalent ones when a new viral infection attack came about. And if that particular viral infection was matching with any of the previously stored sequences, then that was a perfect way for Cas9 to really prevent that threat. And so that's where snippets of these phage DNA had been stored, Cas9 could readily be programmed to slice up corresponding sites. What Jennifer Doudna did was, and her team and collaborators, of course, did was to repurpose this fundamental property for an entirely different purpose. which is to program the Cas9 to cut up other DNA sequences of our choice, viral or not. Just to conclude this, CRISPR is not just a pair of scissors, as it was described in the book, but a Swiss Army knife. It's a single molecular machine. It can achieve remarkably diverse types of gene editing outcomes. It can edit genes, as we know. It can later on was determined to be identified with the ability to do gene knockouts, could do deletions, insertions, inversions. And of course, another extremely important aspect that she touched on towards the end was some of the work that followed their work, which is deactivated CRISPR, which essentially is like a dimmer button or a volume button. You could essentially turn on in high volume the gene expression and control that to turn it on and off, or to dial them up or down, so that it can be more dominant in that sense, to the degree that you want it. So that kind of precise control is even possible with some of the ways in which CRISPR can be applied. So let me stop there, but I hope that gave you a feel for what the journey was that Jennifer Doudna and others preceding her went through that led to that point of discovery.
Thank you so much, Raja. I think, like many have said in the chat, you beautifully recaptured the history of how exactly we reached this point of the discovery by these two women scientists, who, if I'm not mistaken, are the first pair of women scientists to win the Nobel Prize together. So that's another first to their credit. And you really highlighted the tool in a very systematic and structured way. So thanks for doing that. And now with this tool that Raja explained to us, Vinita will turn to what is the task to be accomplished by this tool. So go ahead, Vinita.
Yeah, okay. Can I have the slide? Yeah. So I'll start by reading. And okay. So while I read the book, Crack in Creation, I was reminded of Aldous Huxley's Brave New World. And I thought, are his prophecies actually coming true? Can humans now play God and hence control evolution? Has a Frankenstein been unleashed? These are some questions that came to my mind. As I read the book and got familiarized with this new technology called CRISPR-Cas9, I realized that the benefits of this technology far outstrip its dangers. Genetic scientists Jennifer Doudna and Samuel Sternberg have done a brilliant job of demystifying the science of gene editing while explaining both its benefits and dangers, something which we have already heard both from Smita and Raja before me. Some of the benefits of gene editing using CRISPR technology are to agribusiness. It gives us a chance to create crops that produce more, resist sickness, and are healthier. As Zareen had said, that reducing hunger amongst people when we have more food. Then the citrus industry, for example, has a problem with what's called yellow dragon disease. Using gene editing, they could end the decimation of plantations in Asia that this plague causes. We can also modify our food to be better for us. For example, taking trans fats out of soybean oil. I've just taken some examples from the book, which where Doudna gives a whole lot more and explanations of the positive applications of this technology. Can we have the next slide?
Yeah.
In the livestock industry, gene editing could create new varieties with advantageous traits. For example, getting high yields of lean meat together with low body fat while not affecting the nutritional content and the animal's development, diet, and overall health. For humans, we've got over 7,000 diseases that result from a single gene mutation. All of these could be gone with CRISPR. Even HIV which affects millions around the world, may be preventable with gene editing. It could also find a cure for fatal diseases like muscular dystrophy, sickle cell anemia, and beta thalassemia. This amazing new tool has a chance of eliminating cancer. Because DNA mutations cause cancer, the ability to cut and modify DNA at will might be able to stop it. All this at low cost and ease of use. CRISPR made gene editing available to all scientists. Previous tools were difficult to design and prohibitively expensive. The possibilities this discovery throws up are endless, albeit with a degree of caution and responsibility. I would like to conclude with a quote from the book. The power to control our species' genetic future is awesome and terrifying. Deciding how to handle it may be the biggest challenge we have ever faced. I would just like to say something more, which obviously has happened post our reading of this book, and that was the Nobel Prize, which was recently awarded to a physician, and which was something which was very related to, which I find was related to the Nobel which Doudna received. And that was basically, once again, with reference to DNA. While one technology is looking at, the future of DNA and how it could control evolution, the award of the Nobel Prize for the technology that was just recently awarded was for trying to understand evolution as it has happened before. So I think maybe together with this, we might have something new coming up very soon. So thank you.
Thank you so much, Vinita. I think you elaborated on the task very well by showing what value is added by this CRISPR-Cas9 technology to agriculture or food, to animal life, and of course, to a better quality of life among human beings. And you cautioned yet at the end with your quote that we should keep away from what is terrifying or what is not desirable with this technology. So thank you for that perspective. And with that, we turn to Nidhi to cap all these reflections with some weighty issues like ethics and regulation with this technology, and also the epilogue of the book. So please go ahead, Nidhi.
Okay, thanks, Nidhi. So Zareen asked to focus on chapters 7, 8 and the epilogue.
Nidhi, we cannot hear you very clearly.
Oh, is this better?
Can everyone hear her?
I can hear her.
Is this
It's a bit on the light.
Come closer to the microphone, maybe.
Yeah, I think you're closer.
It's very soft, maybe. We can't hear you.
I've got my headset on, but I guess the headset's not working.
Oh, it's a lot better now.
Yeah, it's a lot better now.
It's working fine. Yeah.
Yeah.
Okay, I'll try and hold on to it and keep it close to my mouth. So Zareen had asked me to focus on chapters 7, 8 and the epilogue. And basically, these, after the rest of the book has the heavy discussion of the technology and how it happens, here Doudna is really reflecting on where we've come and where to go next. So in chapter 7, she talks about this nightmare she has of meeting Adolf Hitler, and her partner has a conversation with a woman who wants to create designer babies. So understandably, they start thinking about, what is it that they have achieved with this technology and where could people take it. She gives a little bit of a history of what happened in the past with Louise Brown, the world's first test tube baby, and Dolly the sheep, and so on. And she points out that historically, there's been very little conflict about using attempts at germline editing for reproductive purposes. But that was more of a hit or complex affair. And now with CRISPR, the power and the scope are much huger, and that is what is giving rise to fear and misgiving. So she makes a very strong case for public discussion. She draws an analogy to nuclear weapons, where she says that there wasn't very much discussion before the technology was developed. There was a lot of discussion after it came into being on how to use it, and she doesn't want that to happen with CRISPR. And she points approvingly to a past pivotal moment in biotechnology, the birth of recombinant DNA, where scientists had moved very proactively and successfully to prevent their work from causing harm. So she makes 4 specific recommendations. One is that the public must be able to get access to scientific knowledge. The second is that the safety profile of CRISPR has to be developed further, much as we've heard on with our previous speakers. The third is that there have to be international meetings with multiple stakeholders, not just scientists, but also religious leaders, disability rights advocates, journalists, and so on. And actually, at this point, she says that there should be a moratorium, hold off on making heritable changes to the human genome. Then in chapter 8, it sort of blows apart, because in 2015, a Chinese scientist, Junju Huang in China, injected CRISPR into 86 human embryos to edit the gene responsible for beta thalassemia. And this set off an international firestorm. In fact, the US Intelligence Committee also got involved and described genome editing as one of the 6 weapons of mass destruction and proliferation that nation states might try to develop at great risk to America. And a whole bunch of philosophers and bioethics people also got in, some saying that it was a moral imperative to aggressively pursue gene editing because it could eradicate gene defects, and others supporting research but not the clinical applications. Basically, Huang from China had released the floodgates. So Doudna and others organized a summit on gene editing in Washington, D.C. in December of 2015. And some of their key takeaways were that germline editing will eventually be safe enough to use in the clinic. She says that the accuracy of CRISPR itself is a problem right now, but it's going to continue to improve in precision and accuracy. And I'm sure she's right, because judging by the level of developments that have occurred in the last 5 years, I'm sure quite soon it's going to be very accurate. A more subtle point that she makes is, will the effects of accurate edits be the ones intended? So the editing becomes accurate, but is that what we want? Some gene edits have secondary effects. For example, editing the CCR5 gene makes you resistant to HIV, but more susceptible to the West Nile virus. But then she goes on to point out that you really shouldn't hold this as a failure, and nor should you wait to correct diseases that cause a lot of problems to people. Because if you're editing to the average of the population, you're probably going to be on the safe side. And I think that's a valid argument. Though later on, we'll get a bit into what others think about the average. But anyway, then she raises the point that just because we can doesn't mean we should. She says the bulk of cases where genetic disease is a risk but not a certainty, should you even get into germline editing for that? And public opinion in the US is pretty divided. Half oppose the idea of using germline editing, even if it is to reduce the risk of disease in a person. Many feel that evolution has been working towards optimizing the human genome for billions of years, and can a small group of human genome tinkerers do better without all sorts of unintended consequences? There are those who think that the human genome is part of a precious evolutionary inheritance, something to be cherished and conserved, and that germline editing changes the very nature of what it means to be human. Another valid issue is that will anyone be able to control how germline editing is used once physicians start using it in the lab? It's a slippery slope from medical necessity to medical enhancements. And Smita and others have also spoken to this. You know, what's necessary, what's an enhancement? And there's a fear that the wealthy could disproportionately benefit from germline editing and that our societal's financial inequality would be transcribed into our genetic code, such as what Aldous Huxley imagined in Brave New World, where we have a society of genetic castes. A more subtle point I thought was, will gene editing exacerbate prejudices against people who fall outside a narrow range of genetic norms? Could it create a less inclusive society, one that pressures everyone to be the same? As previous speakers mentioned, is dwarfism really a problem? Taking it a step further, is being deaf a problem? I mean, how much diversity do we want in our society? How much do we want everyone to be the mean or the average? And again, as both Zareen and Vanita said, few technologies are inherently good or bad. What matters is how we use them. And Doudna's approach to this is informed public discussion. She calls it discussion without dictation. I think theoretically this is very ideal. I'm not quite sure how it happens, but definitely if the public continues to be fearful of gene editing, it's not going to take off to the extent that it can. My opinion is that the genie's out of the bottle. We saw that with nuclear weapons. You could even argue that both Pakistan and India being armed nuclear states has prevented war at a deeper level. So anyway, with gene-line editing too, I think that the worst possible thing that we could do is make it secretive and spottily regulated, by which I mean that the US, let's say, has a lot of regulations, China doesn't. I think that's a recipe for disaster. I think basically keeping it transparent, keeping it open, and continuing to talk is the way to go. And I feel that we've been gifted with an incredible tool. It's got the potential to improve life for all of us. Thanks.
Great. Thank you, Nidhi, for bringing to a close these three reflections with your explanation of the cautionary stance that the authors have taken about what the downsides of this new technology could be. So overall, as I said earlier, the three reflectors have really given us some very precious perspectives on this book. Now we go to Rama to kick off the open session with his thoughts as an expert on this technology. And also I must thank him for recommending Smita Krishnan's name and helping with inviting her to this session. So with that, over to Rama.
Hi everyone. Firstly, thanks Milind and Hiranki for this honor of talking about.
By the way, Rama, where are you at the moment?
Our book.
At my home.
So let me first start off.
Which home, Delhi or New Jersey?
Not cover.
Chacha, this is a recording. I think
It's a recording.
after that.
Yeah, it's a recording.
No, it's Ram Avatar. Rama Avatar.
So this is not
Rama's frozen.
Biology 101.
I mean, what a way to do Dashera.
Biologist, and I don't have that much knowledge. And the second thing is, given the brilliant reflectors, they'll be reflecting upon specifics. I think it's best if I skip that But what I would like to do though is, give you my sense of how to visualize what is happening over here and the implications of it, and potentially bring that together. So the first thing is that, as you can imagine, all of us are just a bundle of cells, and we're not an island. Secondly, all the cells turn into chromosomes, then the chromosomes, which are XX and XY, whether you're male or female, that gets into the gene, and that's where a lot of our genetic information is coded.
What's going on?
And proteins basically make all that also happen. The important thing is that these building blocks are run through the famous double helix, which was discovered by Watson and got the Nobel Prize for it, if I remember correctly. And they really have four broad building blocks in it, which they are A, G, C, and T. And I also, humorously look at something like AT, which is one pair, as like an AZ2 tire, and GC as a general compartment in order to keep my mind clear. It's really fascinating how all these building blocks come together. Now, to visualize this double helix, I'd like us to go back to IMC. We'd go home for the holidays. So imagine you're going from Calcutta to Bombay, for example, and there is the Gitanjali Express and the Rajdhani Express, or just two Rajdhani Expresses or two Gitanjali Expresses, conveying people from Bombay. Assume it's on a monorail. Now, this is in a flat structure. Imagine it's in a vertical structure or a three-dimensional structure and is just twisted. And that is what the double helix is all about. Now, where does Casper and, the Cas protein and CRISPR come? What happens really is that assume that in one of these trains, instead of having, let's say, the Rajdhani carriage, somebody has put in a goods wagon. Now, the train is moving, so you need to have a GPS system to track where it is. Then you need to be able to find a way of identifying where that carriage is, which is wrong, and then snipping, taking it out, and doing all this while all the processes are moving. It's a bit like changing the wheels of a car while it is moving. And that's the brilliance, really, of CRISPR and the Cas protein. We often talk about what each of these proteins do, like Cas1, Cas9, etc. But my simple visualization is, while the train is moving, there is a moving train that first identifies the carriage that is defective, slices the train, extracts that carriage, and then after that, either there is a form of gene repair where the two ends come together by themselves, or a new gene, a correct carriage is then reinserted. And this is really very important for a lot of rare diseases. There are about 400 million odd patients of rare disease. If I remember correctly, about 2/3 of them are girls, and a big chunk, maybe up to 40–50%, don't live beyond the age of 5. And very often there's only one gene that is implicated in that. So imagine the lease of life you get if you can use CRISPR to take out the defective gene and put a good gene instead. The other part of this book that stunned me was the big role that Chinese Academy of Science, etc. are playing. They're far, far ahead. The way I look at it is that if these guys are already taking sheep and making it grow longer hair in order to get better, cashmere sweaters or what have you, imagine if tomorrow they can identify how to make people who are growing old younger in their body. All of a sudden, the so-called demographic dividend that we have in India and the demographic non-dividend that they have because of aging population in China could potentially be addressed. And that may be of interest to us also if you want to kind of live for 150 years. The other part that I found also interesting was the whole aspect of germline editing. And that's the scary part. Because if you can fix the embryo, and then from there it goes into successive generations. Why Mutant Ninja Turtles? We can certainly have mutant men and women, maybe 10 feet tall and with shoulders which are, 112 inches or what have you. So just like chemicals have a chemical weapons treaty, and biological have biological weapons treaty, and we say viruses should not be misused. I think
And we know how well they work.
for us to have some sort of policy framework to ensure that this sort of germline editing and ethical structures are in place and people don't cross a critical line. And with that, I'll let you get back to the rest of the session. And once again, thanks, Milind and Srinivas, for giving me the opportunity to sharing my thoughts. Such a wonderful subject.
Great. Thank
So,
you, Rama.
Milind,
Yeah.
you also had a question on the commercial part of it.
Uh-huh.
And I'll just address that in, kind of take it. So Milind's question was that why is it that stocks like CRISPR, etc., are not doing too well if this is such a fantastic technology?
Yeah, and actually, Rama, I should tell you that this question came from Suresh Divakar. So go
Yeah.
ahead with the answer.
Great. So let me try to address that. So the first is, as I explained in the chat, well, the technology has taken place. The products have not really come into the human arena as much as we would like them to be, because the process is a slow process. It is like CAR T cells, which is chimeric antigen receptor T cells, where the T cell is extracted and then engineered and then infused back in the patient to tackle lymphomas, is an example of it. The second part is that if you take a look at even some of the major companies, there are some people who are trying to create ETFs from all the CRISPR companies. It is still in early stages, but the technology is also in an early stage. The third data point is Gartner Group has got a hype cycle report. And in that hype cycle report, where they pitch things like CRISPR technologies, along with all the other technologies, is really at the peak of inflated expectations, which means there'll be a downward trend where people will try to figure out exactly what can happen, where and how. And as a part of that, biotech companies, whether it is CRISPR with Emmanuel Charpentier or some of the others, will go through their normal pains and pangs of growth. And we need to keep in mind that biotech companies have a slightly different model, except for a few of them. And the model is basically to prove something works in the clinic at the phase 1 level, and then try to sell it to a large pharma who have the resources then to take that molecule into market. So this whole process is still in the 6-7 years arena. But what should be kept in mind is, really, do you get a technology that has, exactly as Nidhi put it together, or as Smita put it in her chart, an application that is so broad-based, that goes from plants to plant genetics, animal genetics, human genetics, and some other areas that we've not even conceived taking place. I'd put my money into an ETF at CRISPR if I wasn't in BlockQ right now.
Okay, thank you, Rama. That is very helpful. We are running a little bit behind time, so we need to make the open session a bit shorter than usual. So just keep to the normal rules, and let's take another 10 minutes maybe for whatever other opinions there are. Go ahead, Ishan.
So what actually struck me when reading the book is that this is also going to increase inequality in the world. I mean, the RNA vaccine itself was created for the rich. And if the developing world didn't have another answer to it, we would all be screwed with the vaccinations between Pfizer and this again, it's exactly what Rama is saying. If somebody will sell it, it's an Israel strategy, create technology and then sell it to the person with the deepest pockets. And when those people monetize it, they'll monetize it first for the extremely wealthy or for governments and rich governments. I think that everybody's talking about the ethical factor, but I think that what was going through my mind was the inequity in the world, the inequality that we're all grappling with is going to go up many-fold if this technology actually, if this becomes an ecosystem.
Can I just respond to that?
Yes, please.
Makes the point as you said, Ishan, about inequality widening, but she says that a lot of medical technology, when it was first introduced, was only accessible to the rich, but very rapidly it kind of became accessible to all. So there'll be a bit of a gap, but she seems to think that very quickly prices will come down to a level where it's accessible to everyone.
No,
Chacha raised a question. I think let us keep a brief Q&A.
Yeah. Okay. So I just have one other amukan. Just one little point, Rama. What about a situation like Wuhan virus happening? Something they're trying and some rogue protein escapes.
Yeah.
So,
So
गड़बड़ हो जाएगी यार.
yes.
Called
So
a
let
gene
me-
drive. That's what a gene drive is.
So, Chacha, if you get me started in China,
something that nobody's proved.
No, if you get me started in China, you may not go to sleep tonight. So I need to keep my passion and obsession to a side.
You
But
have
the
1
fun
minute,
part-
Rama.
Let's
You have 1 minute to
Notice in the
answer
book,
before we move on to
DOD
Sharat Seth.
funded most of Jennifer Doudna's research.
So,
Just,
let me
just
just
a little, and if in the book she talks about DOD funding her research.
Yeah.
So it's probably happening under mines in Utah a lot more than in China.
Yeah. So the point is that whatever.
Rama, I think Sharat wanted to say something.
No, it's okay. It's okay. It's fine.
Sorry,
Go ahead,
go
Rama.
Okay.
No.
Go ahead. Rama,
So there are two parts to it, Chacha, because I want to cover both Ishan and your question in one go. So the first thing is you're exactly right, but it's not, and exactly as Vivi says, it's not just China. I'm sure Russia is doing it. I'm sure people are doing it. And for example, even in India, when the Biological Weapons Treaty was being signed off and ratified, all of a sudden India came up and said that, hey, we also got some labs under control. So everybody will do something, because this is a prisoner's dilemma. The second part is going back to your point of the COVID virus. I share your perspective. I personally think that could be one answer to what happened in the Wuhan virus. But a bigger question is what Ishan is raising, because I'm not too sure I share Jennifer Doudna's point of view that these sort of technologies will be given across. So for example, Novartis makes a drug for $2.3 million, which is Zolgensma, for a very specific spinal muscular atrophy, which is not a genetic basis, but has some things. And there's no way they've reduced the price anywhere around the world. Or their CAR-T therapy, which is CRISPR-driven, is $400,000 a treatment, and it doesn't matter where you are. And the other challenge is when you start getting to GMO drugs, GMO plants, this is genetic plants. If somebody like Monsanto builds it, they will tie in their IP, they will tie in, say, their profit motives. So this is a little messy, and it goes back to the point that Nidhi made in her last slide, which is, there needs to be a much more robust discussion among all stakeholders, and recognizing what Vivi says could well be happening. It's something will happen below the scenes, but do the best to cover equity at least.
let's see if somebody whose voice has not been heard in the open discussion wants to say something, and please limit yourselves to 1 minute at a time. Yeah, go ahead, Arvind.
Got to unmute.
You are on mute, Arvind.
You are on mute.
Yeah, let him complete. I think Nidhi will come back.
Yeah, so this is in response to Anjali's observation about the end of life when people don't want to leave. Actually, I think, there are different types of people, and there are many people who actually want to leave. And they said, "Bahut ho gaya," especially if the quality of life is not as good.
I agree, Arvind, 100%.
I've seen it among relatives, uncles, aunts, the very close fathers, friends wanting to
People
leave.
want to go. I know,
Yeah.
I've seen people. They कहते हैं हमको बख्शो, बहुत हो गया, अब जाना है.
Actually, Sandeep, I've heard people say, one of my elderly relatives actually said that God doesn't love me, he doesn't want to take me.
Exactly. बिल्कुल, 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 बिल्कुल.
Or they say things like, हमारे को भूल गए. The Lord has forgotten us.
इसको भूल गए बिल्कुल.
So I
Okay.
noticed that the Chinese guy, those 3 kids were born in 2018, Lulu and Nana, and he has been released from prison in 2022, in April, I think. And those kids were born, and although it was for beta cell, thalassemia, whatever, sickle cell, and so on, the side effect is improved memory, as they found in mice models. So that is the stuff below the ice.
No, Vivi, I'm
the
sure.
iceberg, yes.
Vivi, I'm sure that it's already happening in the US.
Yeah.
I know about 4 families who always get twins, and they go to the US to have their babies. So kuch na kuch to ho raha hai. I don't. I refuse
Well,
to
it's probably
believe
not germline editing. That is all the other stuff Nidhi talked about. You know, preferential embryo and
That's
all
different, Ishan. That's
No, but what I'm
another
saying
idea.
is,
It's a continuum, right? It's a
I
continuum is
gave
what
it
she's
not as
talking
an
about.
example, but as an analogy. I'm saying
Yeah,
like stuff is happening there, stuff will be happening here as well.
You know, if you have a couple of 100 million, you know,
You'll
And
find
you
some
know,
American.
if you
you'll
think
find some
about it,
American-
if you think about
Okay.
it, if you think about
Just
it,
keep to the rule of 3, please. Don't talk unless 2 other people have spoken. Okay. Go ahead, Anjali.
so if you think about it, when in vitro fertilization started, everybody thought it was a great idea. A lot of people would, infertile people would be able to have children, and it's been a blessing for many. But we didn't see the unintended consequence of one gentleman producing many children.
Right.
And so, you know, so
The doctor.
and now there are many cases and all against that. So I think the unintended consequence cannot actually be thought through completely. And so
That's
maybe
right.
one needs to think about, is there more positives than negatives? And if-
One request. Our guest Saida has not said anything. Saida, would you like to say anything? Comment?
Yes. Thank you, first of all, to let me listen to all this intelligent discussion and amazing speakers. It was really fascinating. And, the book is very interesting and fascinating. As a physician, I do see a lot of things, a lot of diseases where, you kind of like feel like, there is a technology available for this patient. Maybe, they could do something and we have that in our hand, yet, it seems so far away, So it's, again, like they have, it could go into in hands of somebody who can harm. So it's not, there is a lot to be done to regulate, to it's not that it's really not safe. Lot to be done, lot to be decided. So it's I think it's like far-fetched in terms of really coming into our patients' hands. It's in coming to our physicians' hands. It is I don't know.
Thank you, Saida, for your comments. Ajay, we haven't heard your voice. Do you want to say something?
Apart from apologizing for coming 90 minutes late, sorry, I looked at it as 11 a.m. IST instead of 11 a.m. wherever it was, or 11 p.m. wherever it was. So my mistake. Sorry.
That's okay. But do you have anything to contribute?
No, I probably keep my mouth shut, especially having heard the ends of all the discussion. I'm sure there's a lot of intelligent stuff said before I open my mouth.
Yeah,
so
you can always review the recording at your leisure. Okay.
Yep. Thank you.
Yeah. Okay, so I think it's almost 1 o'clock and we should let the East Coasters in the US go to bed. So thanks so much for this.
Thank you so much.
Session, all the reflectors, the reader of the month, and our guest speaker and the final expert. And thanks for attending and your thoughts during the open discussion.
All the
Thanks,
books
guys.
have
Thanks,
been
everybody.
excellent.
Excellent.
care.
Thank you very much. Bye.
And we'll see you in
Bye-bye.
November.
Good night.
Bye.